Thanks. And Richard and Tess, thanks for, okay, let's continue. Yeah, thanks for all the communication. Okay, Zubair has given me a nice challenge before that topic came up. I'll try my best to cover the challenges in the next 20 minutes. We'll leave for some time for questions.
Thank you.
I think what I thought, and there's a ocean of challenges for drug delivery to the eye. So I think maybe I will focus more on the challenges with the delivery of biologics to the eye, because which is the primary focus of Rivana Therapeutics and some of my team members working on various different approaches on delivering biologics, including two of our ESRs. So, okay, let me see. All right, okay. So delivery of biologics, I don't want to bore all you again with the typical barrier problems. So I'm not going to go deep into the physiological barriers, the other barriers in the eye. But as we know that currently different regions of the eye have different barriers that does not allow us to get enough drug through the surface of the eye, particularly biologics. So the only option then remains, which is being used in clinic and as a standard practice, is injecting biologics, solution of biologics directly into the eye, which via the intravitreal injection. It kind of started way back in 1911, where injection was done more to treat retinal detachment by injecting air. But from 1945 onwards, there were injections given for treating some of the diseases in the back of the eye, which kind of escalated over time. And between 97 to 2001, there were roughly 5,000 injections worldwide since the onset of the anti-VEGF molecules, Macugen, for example, in 2006, 2007. There's a significant escalation of injections given in the eye today. So we have, roughly that number definitely has gone up. So roughly 23 million injections are given annually at the end of 2017. That number has significantly gone up since there have been more approved anti-VEGF molecules for treating back of the eye disorders, particularly wet AMD or diabetic retinopathy and so on. So the market has been growing at an exponential rate. It's just predicted to be a $22 billion market for anti-VEGFs alone. Although the anti-VEGFs does not really cure the condition, at least it halts the progression, maintains or increases the visual acuity. It is a lifelong treatment in order to maintain the vision and stop the vision deterioration over time. When given on monthly basis, these biologics work really well, but of course they come with significant burden, a burden of for the patients, to get the injections, and mostly these are elderly patients. There's a lot of social, economic impact for getting the frequent injections. There's an impact on the healthcare center, the doctors, and so on. So it's a complicated process to get monthly injections. And most often patients, some of the patients stay away from getting injections because they're scared of injections, and having that frequently done makes it even more challenging. So as probably everybody here listening to this talk already aware of some of these approved anti-VEGF drugs or biologics, for example, ranibizumab or Lucentis or Aflibercept or Eylea, which are approved anti-VEGF molecules for treating back of the eye disorders. But if you count in the terms of number of injections, Avastin is mostly given because of the price is $50 versus significantly expensive other two molecules. However, there's a growing need of, there's a growing number of injections every year, but some of these molecules are running off their patents. So the companies need to find a ways of extending patents, so that adds the extra mileage. But also, it also solves the problem, the burden of monthly injections. So there are a lot of different companies and research centers or academics are looking into trying to overcome some of these challenges and try to design long-acting drug delivery systems. So I think the next couple of slides will talk more about the challenges of long-acting systems and how where we are currently with different type of systems. And obviously, having frequent injections, given there are problems with it, as you could see on this slide, some of the indications is the patient's compliance, which is obviously very poor. Patients are scared of getting injections into the skin. That's the reason why there's a whole area of microneedles in development work is going on. But getting an injection into the eye, it's scary, leads to pain, discomfort. There's chances of infection, change in IOP, retinal detachment, and so on. Long-acting drug delivery systems, LADDS, offer a lot of advantages and overcome some of these challenges and the burden faced by the patients and also by the society and by the healthcare centers and doctors, and multiple number of issues it can overcome. By improving the therapeutic benefit of these molecules, by reducing the burden. But it also offers the opportunity of that extra mileage for the existing anti-VEGF molecules or the upcoming biosimilar molecules, which want to capture the existing market for anti-VEGF drug molecules. So it does offer a lot of advantages to overcome some of these challenges. We know that from many years, sustained release systems or controlled release systems or long-acting delivery systems does solve some of the problems. But when it comes to the eye, there has been limited work in terms of progress. So as you could see on this slide, 6 of the 8, there are 8 sustained release, or you can even say long-acting based ocular products which have been approved. There are actually 6 of them because Ozurdex or Drista are same principle, like PLG extruded implants. Utic and Eluvin are pretty much the same implant for different indications that use a small molecule fluocinolone. So we are all familiar with the OcuSet and the failure of OcuSet. It was a Alsa's technology that was going to change the paradigm of shift in the way the glaucoma was treated, with pilocarpine as one of the small molecule drug. However, there have been issues of the implant falling off. This is a front of the eye fornix implant that has issues of falling off, not staying in place in many patients. So there were issues that led to the failure of that particular product. Vitricet, which is not in the market anymore, is a surgically implanted device which is similar in principle to Retricet, delivering different drug for different indication. So overall, there have been more or less 3 biodegradable implants in the last 45 years or so. So obviously there has, and that has been done only with small molecules, making this probably more challenging if you have to get a larger molecule like the anti-VEGF that range between 45 to 49,000 Daltons, ranging up to 150,000 Daltons in size. Again, if you look into Google search, you can find intravitreal drug delivery until up to Macugen or even anti-VEGF drugs until 2005. There are very limited number of hits you can probably find. But beyond that, which you can correlate with the drastic increase in the number of intravitreal injections. So if you look in the drug delivery area as well, there's been a significant number of publications from 2006 onwards, leading you to probably 18,000 hits on the literature end. So obviously there's a growing interest among researchers to identify a potential platform to address some of the back of the eye disorders. To date, largely they have been in the small molecule area. The dealing with biologics. Biologics are great molecules, and the top 10 drugs, you probably find top 7 drugs are the biologics with significant revenues. And a lot of companies have been looking into treating diseases by developing new biologics. There's a significant market for those molecules. They are highly expensive. They're complex. They're complex in structure. The stability is a point of concern. At the barriers of the eye, particularly when we try to deliver, I know as an orbital group, we have been working on various different ways of getting the molecules to the back of the eye, other than injecting the material straight into the intravitreal space or vitreous humor. They're large in size, therefore they don't effectively cross the barriers. They're short half-life, and they need a multidisciplinary team to successfully get this molecule across a barrier, create this molecule in the concentration sufficient to treat the condition and also to make it last for longer period of time. Now, before we go into try to formulate long-acting based systems, there are a number of different things that we need to address. Primary thing is the handling of the biologics. Biologics comes as a liquid formulations. You have to either use the liquid formulation as it is, and make your microparticles, gel-based systems, or pre-formed implants and so on, or you got to treat them in order to produce a material that enable you to fabricate your formulation. And that really depends upon what kind of formulation you're making, what loading you're looking for, what kind of target product profile you really have. For example, lyophilization is very common practice. And lyophilization itself is probably the product that is available in the market. For other biologics, they reconstitute, inject. Even with that simple process, there's a significant learning in lyophilization. There's a significant learning in spray drying. The challenge that biologics carry is the level of probably the characterization that you have to do. So in the process of converting a liquid formulation to a dry formulation, a dry powder formulation by lyophilization or spray drying, you need to ensure that you are maintaining the stability of the product or the biologic. The monomer percentage remains high before spray drying or after spray drying, before lyophilization or after lyophilization. And more importantly, you have to maintain its activity. So that's, and that need a multiple number of techniques. Typical techniques as we are familiar, particularly as a PhD student, looking at the formulation side or pre-formulation side, SEC, size exclusion chromatography, HDS phase, and so on. But we need to look beyond those techniques into ELISA, bioactivity assays, looking into neovascular models, and so on. So those are the models that are going to give you the true sense of the activity of these molecules. And the biggest challenge with them is the trend really changes in terms of what concentrations you're handling. Low concentrations, more problems. High concentration, probably less problem. It's not really less problem because you probably have less chances of detecting the issues. And there's a lot of quality control that got to be done with the lyophilized powder or spray dried material. That is also imposes a lot of CMC challenges. How you're going to store them, and the whole aseptic processing of this material that you have to prepare even before you get to the formulation. So that's not the end there yet. You have just started to make the formulation. You have only got the powder. Now you're looking into making a formulation. So there's a whole set of challenges waiting for you before we even get to the point of addressing these challenges by converting the protein into a lyophilized powder or a spray dried powder. Maybe I have just picked up a couple of examples. More often, once we have this powder material, you're either going to make microparticles or nanoparticles. You're going to make gel-based depot-forming, preformed implants, and so on. So I picked up some examples and probably the challenges associated with it. You're all familiar with the microparticle-based systems or particle-based systems. FDA approved PLGA materials back in 1970 has been, I would say, probably a father of biocompatible, biodegradable material, which has been used in significant number of long-acting, sustained release, controlled release products. There are at least 35 to 40 PLG-based products, of which at least 15 over products are based on microparticles alone. And obviously it is working, and for in other areas pretty well. And there are a lot of factors that you have to control: the method of fabrication, the size, the charge, the surface properties, the kind of polymer composition and type of polymer, how you control the degradation of these materials, and so on. So these are the challenges that we need to be cautious of while we're trying to do the pre-formulations and so on. That challenges then apply to any kind of area of application. It's not just the eye, but when it comes to eye, probably the challenges get a bit unique, because here we need to consider a number of different issues that we don't often see in other areas of the body. For example, we are injecting literally particles into the tissue of the eye, and that leads to diffusion of the particles, phototoxicity, adherence to the lens, the charge dependent, size dependent, and so on. There was a nice, interesting study. Probably, I guess this paper from GSK, Polyactiva, probably might be worth $500,000, or probably even more than that. It's a quite interesting work if you get a chance to read. The guys have been using the Polyactiva hydrogel-based microparticles. The target that was 6 months sustained release of a dimeric anti-VEGF molecule. And you can see, as from the release graph here, they have roughly 6 months release within the window of roughly 5 to probably between 25 micrograms per day. Now that's a really huge concentrations, fantastic. And most often we probably get that initial success in the lab scale. The take-home message from that paper was this: the polymer breakdown and the movement of the particles in the eye may limit development of particle-based approaches for sustained release after intravitreal injection. Probably similar is the case that there was another paper from Genentech. They have also seen the safety-related issues with the particles, because you have no control over how the particles behave once you inject and where they end up, leading to histological findings, leading to safety issues. There's also technology from Graybug recently that has had issues causing similar problems, leading to photosensitivity, rise in IOP, and so on. So you need to be cautious about particles. Gel-based systems, it can have phase inversion-based gels, thermoresponsive gels, pH, ionic, etc., etc. They are, again, it has its own set of challenges. What kind of material are you using? For example, phase inverted systems uses NMP as one of the solvent, and that is not an ideal for injecting into the eye. But there's a paper from Genentech that have done the work. It shows fantastic release profiles. That's great. But there's a greater problem in order to control the dimensions of this implant, shape of the implant, the volume of the implant, and so on. Because this is probably not an issue if you have to inject in the tissue, like subcutaneous tissue or in the joints and the cancer tissue. You have less concerns of that. And if you're looking at the scanning electron microscopy images here, the Chang et al.'s group with Raj Gopalan, who has done a lot of work on different formulation, how the implant degradation takes place, the shape change, whether the material is breaking down into chunks, revolving within the eye, how the material, what is the fate of the material. These are all important factors for us to consider. The time lag, one of the significant challenges with most of the depot forming system is the time lag. By the time the implant formed, because of temperature, because of pH, ionic strength, etc., etc., there is a huge burst release happening, ranging from 40, 50, 60% to even 70% in some cases. What kind of volumes you can inject? You can't inject large volumes in the eye. Viscosity is an issue. How you reconstitute, for example, at Rigel technology, there are at least 5 products based on that technology, needs to be mixed at least 100 times before it is injected into the tissue. So there are technical challenges that is not an ideal situation for injecting into the eye. Forces of injections define your needle size and needle diameter. We are limited in terms of needle diameter and needle sizes. Stability of biologics, again, the rate, the faster you inject between the patient to patient variation, the doctor to doctor variation could be significant in terms of shape formation, in terms of burst rates, in terms of degradation profiles. And of course, the sterilization remains an issue with any form of protein biologic loaded formulation. Preformed implants, they are great because you can simply inject them. It is not much prep work you got to do. But to date, with the only two type of implants, which is Ozurdex and Rista, both are hot melt extruded implants injected into the eye, but fabricated at high temperatures, which is not ideal for injecting materials, using materials containing biologics, unless you add some excipients. Excipients, we're all familiar with them. Sugars, very commonly used. But again, they're going to create porous structures. Maybe the physical strength of the implant may not sustain for too long, and it will break down into bigger chunks within the vitreous humor. That might be another problem. Duration, what is the duration of action? For example, the mesh size that you see here in the middle of the slide, this is the Ozurdex degradation picture, SEM image. PLGA, as we know, 50-50, for example, should degrade in 2 months, but that's sustaining even beyond that. So that is a challenge in order to control. And there's a lot more, it happens at the molecular level that we understand very little of, the degradation mechanism that need to be controlled. So duration defines the loading defines the dimension, then the dimension defines the needle size. And swelling, and we don't want material to swell large, otherwise that will lead to issues on non-uniform degradation of fragments of the implants. All this is going to be a significant issue. I added this image here from Grayberg, proclaimed that when you inject the particle-based implants, particle-based systems, they coalesce, they form an implant at the site of injection. Well, it does have its own challenges. For example, how the particles are going to remain there, how long they're going to be there without moving away, what leaches out from them, and how that gets distributed, and what could be the safety concerns. I think one of the biggest challenge probably is the biodegradation. Even though we're going to use biodegradable material, for example, PLGA, which, as we know, degrades into lactic acid, glycolic acid, ultimately leading to carbon dioxide, water. We don't really understand much about it because there's a body of evidence, 55 years of work done on PLGA, yet everything changes depending upon dimension, depending upon the processing, and so on. So these factors need to be critically understood. Degradation defines as the cessation of change, whether erosion defines as this material leaves the system. Just take, for example, Rista, which is a bimetalprost implant, 4 to 6 months long lasting implant. There's a lot of advantages with that, fantastic material technology for sustaining the drug release. But if you look into this paper published last year, the swelling of this implant ranges from 76 to 125%. And at the 12 months, this implant is supposed to release the drug for 4 to 6 months. The degradation is only seen 100% in 6 patients, 25% patient or equal to that or less seen in 26 patients. 100% degradation has been seen in 9 out of 34 patients. So there's a great degree of variability that happens inside the eye. Unfortunately, we don't have a in vitro mechanism that or setup that could correlate, can be easily correlated. So that also leads to the CMC challenges, that also leads to product development challenges and other issues if we don't have a precise control over some of the key product profile parameters. Yeah, development of biodegradable implants is challenging. For example, Ozurdex uses PLGA that was launched back in 2009. Rista, after really 11 years, only now it's released, which also uses PLGA. So there is a gap of almost a decade or more, although it uses the same material, a slightly different issues to address, because now, unlike Ozurdex, which is injected in the back of the eye, Tristar has to inject it in the front of the eye in the aqueous humor or intracameral space. So what I mean to say really is, even though we have 50 years of experience controlling various parameters, for example, I think I did talk about this particular complex issues associated with fabrication of sustained release, control release, or long-acting systems with the PLGA material that we have learned for the 50 years itself is complicated. So making this using biologics seems to be even more challenging. Looks like I'm running out of time. Maybe I'll wrap up in a few minutes. So this is another non-degradable implants. Probably most of you might have come across the port delivery system. That lasts long. Just last week, there's a news that 90% of patients can go to 6 months. That's a Roche port delivery system, or Genentech's port delivery system, which was licensed from Foresight Vision for the treatment of neovascular AMD. So it's a great device. It does does extend the mileage of the molecules, anti-VEGF molecules. However, you need surgery, you need reformulation. It's not the simple injection of Lucentis into these vials. There's a different formulation approach to it. So the reformulation need to be done. So that leads to a regulatory BLA application. It's a non-degradable, obviously. You need experts to do the surgery. Patients, but does patient want to take, patient who does not like to take injections, are they happy to take, have a surgery done in their eye? And then we have a whole set of problems in terms of in our labs and how well we can probably do these studies in terms of what best in vitro setup we can use, and how we can address the problems. Particularly with biologics, it becomes even more complicated. Are you stirring the vial containing the implant, which is releasing biologic? Are you keeping it static? Biologic are not supposed to be touched, because then there is a lot of implications that you got to understand. What concentrations are you operating? What duration are you trying to deliver? So I think these are the different challenges that you need to address. The media, the pH, the temperature, et cetera, et cetera. All these factors are so crucial. I would probably say each of that bullet points is probably a PhD in itself. So it's much more complicated than what we understand. I'll probably skip that slide. In summary, an ideal world, if we could have a self-administering eye drops type of delivery system for delivering biologics, excellent. That's everybody's desire and wish. However, the eye drops are effective in treating anterior segment disorders. Probably they might do well even in small animal models, but probably little to no effect you can see in the retinal disorders in human eyes. For creating a successful long-acting biological-based drug delivery system, there have been a lot of research been done, but there's a very limited success, and there are more failures. Safety remains the biggest concern. I think probably the drug antibodies, polymer degradation, the way the physical structures of this long-acting system degrade is really crucial. Duration versus degradation, ideally you want to balance them out 1-to-1, but that's not, that so far that's not been the case, even with Ozurdex or Tristar. Activity versus efficacy, because that's something is slightly more complicated in terms of you need more different expertise in trying to understand the activity of the proteins and how the efficacy can be tested, and more importantly, maintaining the eye. So different biologics, different challenges. Other options is trying to identify new targets, small molecules, or new biologics. Irrespective of what's happening as of the challenges, the frequent injections of the anti-VEGF in the eye to date remains the standard care of treating several of the back of the eye diseases. And probably that's going to remain for years to come before an ultimate solution is decided. So I think I will probably maybe close up. In order to address some of these issues, we have been working on various different fronts in our Urbana Therapeutics addressing. We have addressed a number of these challenges. We have created systems that can last 6 months, degrade, and so on. So probably I'll leave it there. Thanks to my team, Deepak, Shilpa probably on the call, for their hard work. Yep, happy to take questions. Look like I ran out of time.
Thank you so much, Raj, for a really comprehensive overview and certainly very thought-provoking. We've got a question in the chat, which is, do you think that acidic pH associated with PLGA degradation adversely affects the eye tissues?
Yeah, so I think there is a nice study done by a team. They're actually looking, but let's leave the PLGA aside. Even injecting of simple solutions of biologic in the eye does affect the pH of vitreous humor. And that implications of that is less understood. Now, coming back to PLGA itself, yeah, it does, because the pH drops down to 2. Not an ideal environment where proteins remain stable. pH is oftenly talked as a leading factor for biologic stability, but the other important factor for biologic instability in PLGA matrix is its hydrophobicity. The hydrophobicity leads to aggregation of these proteins that then leads to degradation once the pH drops and so on. So the hydrophobic character of the PLGA, the acidic byproducts, is a double kill for the biologics. And if you're injecting in larger volumes, yeah, it's going to definitely cause a local drop down of pH, as you probably would have seen in the Gelbased technology. For example, when we did a study with Gel technology injected into the skin, we injected larger volume, the whole skin fell off along with the implant because of this acidic nature of the PLGA and degradation over time. The tissue will deteriorate. And I think if you're trying to use smaller materials, smaller volumes, then probably, yeah, it may not affect. But there are other safety concerns with those materials.
Thank you. I guess the other question you've touched on is about the survivability of the medication. So you talked a bit about biologics and handling. And if we're talking about putting these medications in something the patient might actually take out from clinic and apply themselves, what do you think the ways around that might be? Avoiding the kind of problems of the patient shaking the bottle or even just walking around with it?
I'm not sure if I got your question right, Richard. So what you're saying, how patient can interfere with the formulation environment?
So a lot of these protein-based biologic therapies are quite sensitive to degradation.
Oh, yeah. So I think the first thing first, probably the patient will not have the biologics in their hand because they will be given injections. So they probably need to visit the clinic and so on to get these injections. If you are talking about a long-acting system in the injected in the eye containing biologics, obviously they have little to probably no control over that. However, I think the behavior of these biologics may change depending upon the nature of the vitreous humor, because aged patients have a different viscosity of vitreous humor. The distribution doesn't happen, the diffusion doesn't happen effectively, and then either patient doesn't respond or there's a very poor response. There's already seen in the cases with Avastin, for example, 50% patient doesn't respond. So there will be issues with those issues will be same as that you see with sustained release system versus a solution. But yeah, I think the challenge always remains, in terms of development, how you could avoid the stirring and shaking to induce issues such as aggregates formation and other issues.
Thank you very much for a really stimulating talk. It certainly made me think a bit more about how we deliver drugs. Our next speaker, do we have on the call Dr. El-Sher?
Oh,
yes. Thank you very much. I'll hand over to Dr. El-Sher from Kingston University.